for the introduction as well. So yeah, as was said, I'm a material scientist. I'm a chemist in the University of Liverpool, and in recent years, I've got a long sort of history of making both therapeutic and diagnostic nanomedicines. And in the last few years, my research sort of focus has kind of pivoted away from sort of MRI and infectious disease research towards well, it's not pivoted. I've always been interested in it, but in the last few years, it's kind of accelerated, I suppose, into this technique called magnetic particle imaging, which is quite a relatively new technique, and it's becoming more and more known as amongst sort of the imaging community as the years go by. So magnetic particle imaging. So the reason, obviously, why I'm bringing this up as well is because, and why my interest in this has kind of has gone towards this is at the University of Liverpool, we now have a preclinical imager, the MPI imager, and that's our one there in that image on the right hand side for Magnetic Insight. It's the Momentum MPI scanner. How MPI works, I'll go into the next few slides, but it's a diagnostic imaging technique which detects nanomolar concentrations of spion traces. So they are superparamagnetic iron oxide nanoparticles, and it offers real-time quantified in vivo images with no background signal because the body has no magnetic material present. This here on the right-hand side, that is the MPI imager. It's the Momentum scanner. At the time of installation, it was the 12th Magnetic Insight Momentum scanner in the world. There are other companies that make them, so the Bruker as well. And I think since we've had it installed, I think there's a couple more that have kind of popped up. This here is the Hyper. So that, in addition to the magnetic particle imager, we have the hyper-focused hyperthermia platform as well. And that's one of only a few in the world. I'm not actually sure exactly how many there are of these scanners. So what the Hyper does is focused magnetic fluid hyperthermia, and it works sort of it's not too dissimilar to how MPI works, but it's much higher frequencies to cause heating. The scanner comes with software for tracer development, which is obviously, as a material scientist, that's what I'm very interested in. So tracer development and optimization. So it's relaxometry, basically, that comes part of the scanner. And it comes with another technique called the Momentum SDK, so for image reconstruction and all this kind of stuff. So it's quite a powerful tool. Next slide. So how does MPI work? So MPI employs a gradient magnetic field to define a local zero field region of space, termed the field-free point. So essentially what you do, in very sort of basic terms, you get it's like having two magnets kind of pushed together. So if you have the north and north of two magnets sort of pushed together, those field lines would repel, creating this strong sort of gradient field. And within that field, you have an area of within the field lines where there is no magnetization. And that's what the field-free point is. So you have strong magnetization in those field lines, but in that area there, you have no magnetization at all. Okay, so if you are a magnetic particle sitting in the field lines, you would be fully saturated, fully magnetized. So if I get on my next bit, you'd be fully saturated and fully magnetized. And if you are in the field lines on the other side, you'd be fully saturated as well. But as the particle either passes through or the field-free region is swept over the particles, they would immediately lose their magnetization and go to zero. So what is done in MPI is across the field-free point, you apply a drive field, okay? So a field is sort of an alternating drive field is applied, which therefore the only spions that would respond to that sinusoidal drive field are the spion in the field-free point, because the ones in the field lines are completely saturated, okay? So you drive your drive field across the field-free point, and you can see in this sort of schematic I put at the bottom, as they hit the ones in the field-free point, they oscillate, but the ones in the gradient in the field lines, they stay completely saturated, and you get this sort of oscillation sort of response that your particle is doing as it's kind of fluctuating backwards and forwards as that drive field kind of goes, and that is taken And it's a derivation of what you call the Langevin magnetization curve. So that's what this is. That's the Langevin magnetization curve. And the derivation of that is what gives you your image, which is interpreted as what you call the point spread function. Okay, so that's how MPI kind of works. Just to quickly comment that I'm not a physicist, I'm a materials chemist. So that's if anybody has more sort of detailed, very physics questions, I might be a little bit sort of difficult to kind of go exactly into all the ins and outs of everything. But in layman's terms, that's basically what it does. You create by creating large gradient magnetic fields, you have a point where your spine is not saturated. You oscillate the field against that. So only in that region your particle oscillates, and you from that it's picked up by the receiver coils and you generate an image. In terms of MPI versus other techniques, I've just put a few up there. So MPI, this is MPI at the top. So yeah, it works by the nonlinear magnetization response to the sinusoidal drive field. It has a spatial resolution at the moment of about 1 millimeter. The acquisition time can be quite fast. And it's because there is no signal to noise, because there is no magnetic material in the body, it can detect quite low concentrations of that tracer. There's limited harm to the body. There are some potential biological effects of magnetic fields on people, but not at the field strengths of the MPI. I will also just quickly comment that MPI is still only preclinical as well, so it's not quite yet known what it could do if you had a full body scanner. When you compare this to the sort of the obvious, when people kind of think of magnetic particle imaging, the obvious one to sort of compare it to is magnetic resonance imaging, even though they work in different ways. So MPI looks at the tracer directly, whereas MRI works on, it's essentially, if people know what NMR experiments are, so nuclear magnetic resonance, it's a proton NMR experiment on the body. So you visualize the magnetic response of water protons in various different tissues, and that can give you contrast. And again, it's a resolution of about 1 millimeter. They take much longer than an MPI because there's so much water in the body, you kind of get a lot of background signal, and you have to do long scans in order to have your contrast. For that, you can add a contrast agent, and they're typically gadolinium, which is what I did a lot of my previous work on. But gadolinium contrast agents themselves have some possible toxicity and all this kind of stuff. But it's yeah, you can sort of see that MPI and MRI are similar in the sense that they create a magnetic field, but they're quite different in how the applications there. One comment to quickly say is that MPI, because there's no magnetic material in the body, you don't actually get any anatomical information. You only see the spion. So in principle, you would just see a floating spion in the middle of black space. So it will always need to be accompanied by another imaging technique, and very often that's CT. CT works by X-ray. It's got a very good resolution. It's very fast, but it does have the possibility of harm through ionizing radiation, so X-ray radiation. So it's not great in a sense in that regard. But because they're so quick, you kind of you minimize that sort of. My screen. Oh no, it's okay. And then obviously, just very quickly to mention other techniques, so positron emission tomography and SPECT, they use gamma radiation. Resolution's still pretty good. They take minutes, but obviously you're using an ionizing radiation. So MPI has its advantages against other techniques in terms of, limited harm. You're directly looking at the tracer, and you can quantify that tracer. I suppose it has the limitation that you will always have to use a second technique to accompany it for anatomical information. And you're limited at the moment to only preclinical. There's a lot of work going to try and make clinical strength MPI. So focusing on the spion to begin with, so superbiomagnetic ion oxide nanoparticle spion tracers. So the resolution and your image intensity derives from a combination of the Brownian and Néel response in the presence of that sinusoidal field. Okay, so Néel, and it's a combination of the two. So that's your total relaxation. Néel relaxation is kind of the magnetic moment relaxation. It's like swapping north with south, so to speak. It's the internal rearrangement. So as that kind of rotates and oscillates in that sort of oscillating field, whereas Brownian relaxation is kind of the whole particle rotating. So you can see that the entire thing kind of moves. So it's based on sort of diffusion and all this kind of stuff. But those two processes work sort of in combination to give you your overall relaxation. And if you break down, without going into lots of the detail of these equations, um, nail relaxation basically it's dominated by the anisotropy constant of the particle and the core volume. It's more related to the actual particle versus Brownian relaxation. Both of them are related to the particle. They both matter what the particle is. It's just that it's more dominated by things like the viscosity. It's because it's Brownian, it's based on diffusion, so it's more dominated by viscosity and the hydrodynamic volume. In summary, Néel dominates for smaller particles at higher frequencies, and Brownian dominates for larger particles at lower frequencies, with a theoretical optimal spion core size of about 25 to 30 nanometer. Kind of, I should have put the reference for this actually, but it's kind of sort of theoretically determined to be about 25, 30 nanometer is the ideal size of your magnetic core. And this is how these things sort of manifest. So that previous slide before showing the derivation of your peak, you create these what you call point spread functions. So these are the images at the bottom, and this is a derived point spread function that you can get either from relaxometry or from the image. And this one on the left-hand side, that there is showing the sensitivity of two particles. So particle A clearly has a very good response. So it's much more sensitive than particle B. And on the right-hand side, how you interpret resolution is to normalize these two curves and look at the width of the curve at the full width half maximum. So here you can see the wider it is, the less resolved, and you can see that in kind of the images that you have here. Although I feel like particle A, sorry, I got it the wrong way around. Particle A is the least sensitive and less resolved, and you can see that in the images at the bottom. So it's a combination of Brownian and Néel relaxation. When I very first saw somebody present MPI, it was Steve Connolly that was that's from Berkeley, and he's one of the kind of the pioneers, I guess, of MPI. Straight away, I was looking at this sort of stuff and thinking of all the various different, as a chemist, all the various different sort of chemistry things that can be done that can exploit those two sort of processes, and that's how I kind of started to get quite interested in what MPI actually might work. So just to quickly go on about superparamagnetism. So one major advantage of MPI over MRI is magnetic moments of atoms are massive by comparison to a nuclear magneton. It's about 1,000 times higher. So in other words, looking at the magnetic signal response of an atom versus just the nucleus of a proton is significantly higher. So you're automatically going to get a much stronger signal. Superparamagnetism is where you have the immediate and complete loss of magnetization in the absence of a magnetic field. Okay, so I've drawn here the difference between superparamagnetism and ferromagnetism. So if you sort of apply a magnetic field, your particle, the way I always use this as an analogy is think of a compass pointing north. If you apply a magnetic field, the compass will swing straight to the where the magnet is, and that's what's going on. Your particles, the magnetization swings to the magnetic field, swings to where you've got your magnet. You take the magnet away, it'll swing back. And in a superparamagnetic particle, it will immediately swing back and become non-magnetized and then swing the other way if you put the magnet on the other side. In ferromagnetism, it will retain some of that magnetic behavior. Okay, so instead of immediately demagnetizing, so like a fridge magnet, for example, it maintains its magnetic property. You kind of have to pull the magnetization away, which is why you get this curve that kind of looks like this in one of these hysteresis loops. Okay, why that matters? So it's superparamagnets are very strong. They're very high like this. But why that matters is, in principle, what you would want is in that field-free point. So the way I sort of think of it is you're oscillating this field. You would want to rapidly saturate, so it's in this state, and then rapidly desaturate so that it's not kind of oscillating in that sort of, you don't split that kind of signal and blur your signal. So you want your magnetization to be strong enough to be fully saturated in the field strength that you have as your scanner, and immediately desaturate in line with the frequency of the oscillating field. So in other words, if that field is going so fast and this does not fully saturate quick enough, so it's not able to keep up with the oscillating field, you wouldn't fully saturate your particle. There would be some kind of resonance or the remnant, sorry, of your particle, and you wouldn't get a strong signal. So ideally, what you want is a very responsive particle, that magnetization as high as you possibly can, because that signal intensity is related to the height of your magnetization, and it rapidly saturates and desaturates as fast as possible. You don't want the ones here, and you only want it to respond to the sinusoidal field in that point as well. You don't want it to be sort of here kind of responding, if that makes sense. So that's what you want. You want a high magnet, as much magnetization as you can, and a rapid fluctuation of that of its magnetization as the dry field sort of passes over. So when you start to design tracers, there are these are some of the commercial ones that are kind of there. You have multi-core tracers, or you have single core tracers, and very often they have to be within some kind of biological sort of protecting sort of shell. Commercial spions here, so I've listed a few, and they're different sort of properties. So you've got the bimodal Vivotrax, which is the one that Magnetic Insight provides. So that's one that the scanner is kind of built around, is Vivotrax. With carboxydextran, and then you have these nanoflowers, Cinemag, again with dextran shells. You have these larger particles, MPIOs, that are quite often surrounded by polystyrene, so they're very, very stable, and they're much larger, so they have a very strong magnetization. And then you've got the Farahim at the bottom, which again is a smaller core carboxydextran shell. And here is, in this paper here, they compared them. So you can see that very strong signal comes from Cinemag, followed by MPIO. I think I showed in the previous slide that anisotropy has a large effect on the magnetic signal, the Néel response to the magnetic signal. And if you've got a nanoflower, you obviously have quite a anisotropic particle. So that's probably why that has such a strong signal. And then you have your sort of, by comparison, not as strong. So the one that actually is provided by Magnetic Insight relative to others is not necessarily one of the best ones. And here's all those peaks normalized to show you the resolution of each one. There's two ways to develop spions. Well, I mean, there's many ways to develop spions, but there's two sort of commonly used synthetic methods. One is thermal decomposition, and one is co-precipitation. And they both have their advantages and disadvantages in sort of doing stuff. Thermal decomposition requires organic solvents, massively high temperatures. It's quite hard to scale up, but you get fantastic particles. Thermal co-precipitation, on the other hand, is much quicker to do. It's basically stirring, and not room temperature, it's about 60 degree, and you get very nice particles, but they're not so crystalline, and they're not by comparison really that great, but they're always just done in water with biologically relevant stabilizers. And here is a couple of examples that I thought I'd just sort of put up. So this is a thermal decomposition particle. They're varying the oxidation of the particle, and you can see how strong that magnetizes, and how quickly on this particular one that was made here using thermal decomposition, when they add oxygen and they completely make their core just single crystal, single type of core, you get this very strong magnetic signal and very quick response. And this is the signal intensity of that's just the derivation of that sort of sweep. So that's turning that into these point spread functions. And here on the other side, we have a group that this is the references down here. We have a group that's made various different shapes and various different clustering. And so shape, anisotropy, has an effect on particle signal, and also the proximity of particles to one another will have a cooperative kind of effect on the magnetic signal that is generated. Without going into detail of exactly what was in all of these papers, the point that I'm sort of making here is you really do change your particle property by synthetic method, by the process that first of all, the process of synthesis, but also the manner in which these particles kind of interact with one another, and the shell coatings that you can kind of have with them. So from a materials perspective, it's fantastic. The other thing to also say is MPI as a technique, it's tracer driven. So there's no the scanner, for example, that we have is built around vivo tracks. Quite often, if you develop a new particle, it might not actually be suitable for the scanner that you're actually using. So in principle, you build, and that's why there are many groups building other scanners because they're more suitable to a particle that they're actually developing. So the surface chemistry that you can do, so obviously you work on the core, but really the surface chemistry is, if not anything, it's actually from a bio perspective, it's much more important because this is the bit that tells you where it's going to distribute in the body. And also, if you want to have some kind of functionality to your particles, this is where you can introduce that functionality. So you can have polymers that might respond to something, or you could have targeting vectors like I've listed here. So you have, antibodies or aptamers or peptides or just, molecules so that they target to places. So you can control particle stability and biodistribution, and this is the research that I kind of do by the varying the sort of surface coating just by simply varying the polymer or the stabilizer type. So polysaccharides or polymers, you could put them in silica, so they become much more inert. That will obviously have bio implications as well. And yeah, we work a lot on amphiphilic polymers where we try to control the sort of the tuning of these particles, which I guess I'll go into a little bit more when I talk about my own research a little bit later on. But from a chemist perspective, this is absolutely fantastic because you can do so many things. You also can use them as drug delivery, and because MPI allows, and I've got a couple of examples of this, because MPI allows for quantitative real-time imaging. You can actually quantify and look at exactly it's genuine image guided therapy. You can look at exactly where your particle is going and where it's depositing drug. Just to quickly remind that we do also have the hyper as well. So hyperthermia works by heating particles. So there is a lot of potential here, not only for MPI, but also, for example, these drugs could be released by heat sensitivity. So there is the theranostic kind of side as well. So you could be developing particles that give you the ideal MPI signal, but also the ideal hyperthermia response too. And trying to tune the two is really interesting. So there's further spine investigation, and heat sensitive composites can kind of come with that. And finally, as another just sort of thing, it's not really related, I suppose, to surface chemistry, but it's just a reminder to anybody that might be interested in going into MPI research. It will always need a second imaging modality. So there is an enormous potential for multimodal imaging agents and imaging science for image reconstruction. So you can combine your MPI with CT or MRI or something else. So from a material and device perspective, it's actually quite powerful in terms of research opportunity for people who are interested in doing stuff. So obviously there's bio applications, but there's an awful lot of just material development that can be done as well. So applications for MPI, there are quite a few. I mean, I've listed a few to try and make this sort of diagram here. So it's not very graphically great. It's it's an okay diagram. There's better ones in different papers. But I just wanted to basically point out that there are many different things that you could use MPI actually for. And the thing that I'll focus on is, I've mentioned it a few times, the quantification aspect of MPI that you actually have. So just the reminder that detection is spatially resolved to the field-free point. Okay, so you only see the particles within that field-free point. So that's you're sort of defining that sort of image voxel by creating the strong field gradients that you actually have. So you only see the particles here. And the MPI signal is linearly dependent on the magnetic moment within that field-free point. So therefore, spine concentration can be calculated, can be determined. And I've just put this from this person's paper down at the bottom. It just demonstrates that's a calibration that has been done where the iron content has been calculated, and you can see this linear increase from iron content that is present in the sample, but also presumably present within the field-free points. And you can see how that actually is. And that's a really powerful aspect of MPI because you get no background signal and you can quantify your particle. So it lends itself to things like cell tracking, drug delivery, as I was mentioning, and all these various different things, because you can now quantify stuff versus seeing an image from just contrast perspective that you can infer quantification from, but this literally quantifies stuff. So MPI offers quantified and real-time monitoring with no background signal. So in that regard, it's very powerful. I just go into a few examples. So here are a few examples of one of the most common uses for MPI, which is cell tracking. So cell tracking obviously really exploits the idea of being able to quantify the magnetic component that you have, and therefore quantify cells that are migrating and whatever cell tracking study it is that you want to do. It's a bit wordy, this slide. I've put the references at the top, but this one here is what they say was the first in vivo tracking of stem cells using MPI. And essentially what they did was they labeled these mesenchymal stem cells with Reservist, just with the standard, or Reservist, which is also Vivotrax, just with the standard agent that most people are using for MPI. And over time, they see the clearance, and they can quantify the clearance of the particles from the distribution that they have. So results indicate that labeled cells immediately trap in the lungs, and then over time they clear to the liver. This is versus the Reservist, so they did the comparison within the cell and just Reservist on its own, which are immediately taken up by the liver and the spleen. So they used it to see that you can actually do it, so to speak. And in this study on the right-hand side, it's a cell tracking study where they've labeled it's using much larger particles now, so MPIOs, labeling murine mammary carcinoma cells. And they are doing a study where they are seeing, again, the depletion over time, presumably as the cell, there is cell death, and the tumor is decreasing, and you can therefore see the depletion of those tumor manifesting in the loss of the sort of magnetic particles that are present within there as the tumor goes away. So it's very, very useful for both knowing where particles are going quantitatively and also monitoring their presence over time. It's really, really powerful. So those images there are the MPI is the dot, so to speak, so the bright spot, but the anatomical would be from a CT scan. MPI can also obviously be used in as just tracers, non-labeled for cancer imaging. I mean, it can be used for all sorts, but I'm just focusing on the ones that I find interesting myself. So here on the left-hand side, this was a study where they used non-specific spion. I can't remember which spions these were, actually, I should have written that down, but they got them from a company, just non-specific spions, not labeled with any kind of targeting agent. Oh, there they are, the LS008, so commercial spions that just have a code. Distributed into the animal, and you can see what they did was they basically were using MPI to demonstrate that you can monitor the effectiveness of the EPR effect, the enhanced permeability and retention effect, just from these long circulating spions filtering into those tumors. So it demonstrated in this paper, the use of MPI for direct and linear quantification of that particular particle's EPR dynamics. So just on its own, to understand biodistribution of new particles, it's very useful. But on the right-hand side, it's a little bit more of an advanced study where they have got labeled particles now using this lactoferrin functionalized particles. And again, it's kind of the same principle, injected into the mouse, and the particles selectively accumulate within the tumor, and you can therefore see, first of all, the effectiveness of that particle in its targeting, and also you can image where tumors are as well. So that's a really useful study. And this last one, I really like this paper. This is it's very linked to the research I've done over the last few years. So this one here is MPI for drug delivery. So it's a very interesting paper. The reference is at the bottom. And essentially what they did was they got PLGA doxorubicin loaded nanocarriers that contain spion. So that's what that picture is there. So you can see they've got this sort of everything particle, so to speak. You've got a nanocarrier that carries both your imaging agent, your spion, and your therapeutic agent, doxorubicin. So it's a theranostic particle. And over time, they were able to image over this 48-hour period the gradual accumulation of this particle in the tumor. So you can see that the MPI particle, this large particle, did filter into that tumor really well. I've only shown this graph, but there's another graph as well that demonstrates that as that nanocarrier broke down, simultaneously you get the rise of the doxorubicin in the local area and the release of the particles. And if you remember back from the whole Brownian Néel discussion before, how these work, in principle, these get brighter because they're able to move easier. So that's what happened. You saw an increase in your MPI signal at the same time as your release of the drug, and that was demonstrated in the tumor depletion of the. You can see the increase of the signal there and the loss of the tumor as well as the therapy kind of went on. So it was quite an interesting study, this one. And again, just to quickly remind, hyperthermia. So just to go into slightly more detail, hyperthermia is the use of, so basically it's the use of oscillating magnetic fields to create heat within your spion. And why this is really useful is by generating that field gradient, you can create that oscillating field at that very focused point. So you can really, in this system, you can really focus where your hyperthermia occurs. So you can really have your hyperthermia right on where the magnetic particles actually are going in the site where you want them to be. So in other words, if they're a spion that are sort of distributed all over the place, they're in other tissue, and you just oscillated your field, all of those spion would be heated, and therefore you would have off target damage. With this system, you can focus it exactly where your tumor is or where the accumulation of your particle is, and it's really useful in that regard. The frequency of that drive field for an image is about 45 kilohertz. The frequency of the drive field for magnetic fluid hyperthermia is much higher. It's about 350. So that's the only thing that's a bit different, is you do need much higher frequencies in order for that to actually generate the heat that you need. And in this study, again, a very interesting study that was done. This is using the same, so it's not our system, but it's the same system over in America. And here it just demonstrates exactly that. So they got their spions. They are accumulating in the two regions here. So they've got two tumor where they have down at the lower end of the animal and one higher up. And you see the localization using MPI. And basically what they did was they heated only at the presence of the one tumor. Okay, so they targeted it specifically just at that one site. And then over time, using bioluminescence, they saw the loss of that tumor specifically, but not the loss of the other tumor. So basically, the point of the study was to show how focused this can be, because obviously a mouse is, they're really close to one another. They're really, on a mouse, that's very, very close. So that is really, really precise and really, really useful. If you had a drug loaded in that particle as well, you would deposit that drug in that location too. So that would be a future theranostic sort of idea. So to just go into a bit of my own research, so just to say, for a long time, I've worked on nanomedicines for both HIV and cancer for drug delivery. Prior to that, I was working on gadolinium-based, excuse me, functional sort of MRI contrast agents, and it was about 5 years ago now, I won a fellowship in order to essentially combine the two. So I now work on theranostic particles that are essentially what get referred to as polymer cargo, sort of cargo delivery kind of nanocarriers. And this is kind of the schematic that I always sort of draw. So you have the polymers basically that contain your cargo, which is it's similar to the previous paper, where you have your iron oxide and also some drug as well. That's kind of where I kind of like to go. And I've over the years I've got a couple of papers on this, and we've got some patents as well where we've created different systems for basically making these materials. What's not been able to be done until recently is the actual MPI analysis of these things and the hyperthermia analysis of these things. So that's what now I'm very excited about being able to start doing. So just to go into the first method, we're doing it via both methods. This is the co-precipitation, but we also use thermal decomposition. So this is polymer-directed spion synthesis. So we are now investigating the effect of use of amphiphilic block AB block copolymers, so polyethylene glycol with methacrylate polymers made by a method called ATRP, which is atom transfer radical polymerization. It's a very controlled method of making linear and also branched polymers. And we've made a whole library. We've got tons of these sort of polymers with various different groups. And we are looking at the impact of polymer composition on the overall structure of your particle. So I refer to that large thing as what it was on the previous slide as the blueberry muffin. The blueberry being your iron oxides and the muffin being your sort of polymer kind of surrounding them. So in this, we're looking to see what the effect of the polymers have on the muffin, so to speak, the size distribution and its behavior, and also the impact that has on the growth of those particles, the blueberries, so to speak, inside your blueberry muffin. And we are seeing, I've just drawn an indicative XRD of these things, but we are seeing that, first of all, we are able to change the hydrodynamic diameter by varying the composition of the particles, and it is having an impact on the core size that you have inside your sample, which is therefore, on the next slide, having an impact on the MPI data that we're seeing. So each of these, this is just to show what this actually is. So each of these contains the same amount of iron. These are various different samples, various different compositions, different polymers, different concentration of polymer relative to synthesis and all this kind of stuff. And it's showing that what we're making in our libraries of study is we're able to tune the ability of these to respond, the effect, how good they are as MPI agents by varying the composition. So we're showing structural change from the polymer perspective on how this is actually affecting your MPI signal. What is it? We've not been able yet to do any in vivo with this, but we're about to start doing it. And what is actually kind of interesting is, although samples may have a lower MPI signal when you're doing an in vitro relaxometry study, they might accumulate different because we're able to also adjust hydrodynamic diameter. So although the signal might be less, it might be, you can have the world's best MPI agent in a vial sat on your bench. If it doesn't give a signal in the location you want it to be, and then you don't have an agent. So that's, although this, for example, this one might be low here, it might have better accumulation. And that's what we're now about to start looking at with these samples. But it was it's quite a long sort of process that we're kind of going on, and it builds on all of my previous stuff of combining these with drug carriers and combining these with other things. And we also would like to look at the effects of heat generation as we kind of go through these particles as well. But this is using co-precipitation as a method. The other method that I mentioned is thermal decomposition. It's a much more intense sort of technique. It requires, things like sodium oleate, oleic acid, which are very high boiling point materials. You raise temperatures to 300 degrees. It has to be very controlled. It's not easy to scale, but you make absolutely fantastic particles. And what we did in this study was, it's my postdoc. She's absolutely brilliant. She makes so many sizes and shapes and all sorts of stuff by varying the different parameters of this synthetic method. And in this particular study, what we have is large particles, small particles, and poorly dispersed particles. And on the one at the bottom as well, we have exactly the same principle, but with cubes. So these are spheres, these are cubes, large cubes, small cubes, and poorly dispersed cubes. So in principle, what we wanted to see was, we're investigating anisotropy. In principle, these would be too small, probably, to fully saturate. They wouldn't be strong enough to fully saturate in the system. So they would be a poor signal, and these would be a large signal. And what we want to know is, if you make a polydispersed sample, can you correct for that? That's essentially what this study actually was. And is that correction greater or worse for when you've got a cube versus a sphere? That's essentially what we're kind of doing. But this is just one aspect of a much larger study, obviously, that we're kind of doing. And that's what we saw. We see exactly what we predicted. We saw very good signals for your large particles, really bad signals for your small particles by comparison, but much worse for spheres than you saw with cubes. So that's probably an anisotropy effect. What was interesting was, although these were very polydispersed, clearly the larger particle has a dominant effect on your signal, because that contains both this and this, but you're still seeing quite a nicely resolved image. So you could potentially, and this is just the same data plotted as a normalized curve, so that you can see the resolutions of difference. So that's what we kind of are starting to look at. Can we first of all optimize thermal decomposition, or just look at thermal decomposition as a technique? At Liverpool, we have next to the Department of Chemistry, it's called the Materials Innovation Factory, and it's all about scale up. It's all about automated systems for large developments of materials. I haven't, as I imagine I'm talking to a lot of academics in the room, as you probably all appreciate, it's difficult to get money and justify things and all that kind of stuff. But I'm really trying to win some funds so we could try and develop the scale up of magnetic particles optimized for MPI, because hypothetically, if MPI starts to become such a technique, it needs the tracer, and therefore it's not like MRI. You can run MRI in principle without a contrast agent. MPI, you cannot, if you don't have a contrast, if you don't have the tracer, you don't have the image. So there could be a need for larger scale up of these sorts of things. So we're approaching both and trying to win some funds. So in summary, I hope it's come across that I've talked about the various different opportunities in terms of material science and synthetic chemistry, but from an MPI perspective, and how the signal, I thought I'd explain a bit about how the signal kind of is generated and contextualize that sort of material science that kind of comes out of that. So you can sort of understand that it's the Brownian and AL, and therefore you optimize your signal by size, shape, and multi-core and all this kind of stuff. But for those interested in bio applications, obviously surface chemistry and biotargeting are really interesting. And then obviously you've got your composite materials, theranostics, hyperthermia, and all this kind of stuff. So it's a massively wide field that a lot of, I haven't even talked about image reconstruction. If you're a mathematical image reconstruction scientist, you'd find this extremely interesting from that perspective as well. So just to say thanks to, I love these sort of slides because it's almost like thanks to everybody I've ever met. But these are the people that are directly in my group over the years. They're not all there now, but this is over the years. These are people in my group. These are people at Liverpool that I interact with specifically with MPI, but these are just some other people in Liverpool that I interact with just generally across all the science. And we had a research project we still have a collaboration ongoing with Paula Foster over in Ontario to work on some application, because she's got the scanner as well. And these are all the funding bodies that have helped us. I'll just quickly highlight this as well. This is a cost action that recently has just launched. If anybody is in, it's very specifically for MPI and theranostics. So if anybody is interested in having a look at that, that's the info right there. And these are all the other funding bodies. And that's it. That's the end of me. I hope that was okay. Yeah. Thank you.